great to have you all here. Great to see some new faces to the AI Institute seminar. And hey, everybody online. So it's a real pleasure to have Charlie back, Charlie Fieseler, who was a PhD student here. And he was in the physics department, and he worked with me, and we did some work on neuroscience. We were working at C. elegans even back then.
Yeah.
And when he graduated, he took a position as a postdoc in Vienna to work with Manuel Zimmer. And Manuel Zimmer wrote, he's one of the just absolute leaders in the field of worms. C. elegans worms, actually one of the first to actually image hundreds of, so the C. elegans only has 302 neurons, but he was the first to image all of these, well, not all of them, but about 100, while this thing was alive, lighting it up. And so it's just amazing data collection process that actually transformed the field. Manuel, I think, set the standard for everything that came after that. And then it allowed people to think, actually, we can do this. And now, today, really because of that contribution to that group, you can do whole brain imaging for freely swimming worms, tons of the neurons. And it's not like you said, I imaged 100 out of 1,000,000. It's like you're getting most of it.
Yeah.
And so Charlie's been there, and I'm super excited to see this because essentially, I would say that Manuel doesn't publish that often.
No.
But when he does, you should listen.
Yeah.
Okay. And so what I'm excited for you guys to see today is this is the next
fire,
next
the
big
cannon.
hit.
Yeah.
The next big hit out of the group. So Charlie's here. Last comment about Charlie. When Charlie graduated, it was a sad day. Because Charlie everybody missed Charlie. Because if you meet Charlie, he's just so much enthusiasm, so much support for the group as a group member. And so a lot of people missed having that energy around, right? He brought people together, and I miss Charlie being around, because if you need a little burst of sunshine, like today, we don't need it. There's a lot of days to see how you need a burst of sunshine. All you got to do is go see Charlie. And I was like, you didn't even get sunburned. So it was great. So Charlie,
I'm not trying to kill you like all that UV stuff. Yeah. Okay. Well, thank you so much for the very kind introduction, and thanks for inviting me here to give this talk. I'm going to be using this laser pointer here, so I'll try to also say verbally what I'm focusing on for all you guys in the Zoom call. So sorry that you will miss this wonderful laser technology. And also I just got here from Europe yesterday, so if I'm speaking quickly or otherwise not understandable, please stop me and ask a question, and let's clarify things. So as Nathan said, this is kind of the first paper in a couple years from the Zimmer lab, and it is now on bioRxiv. It's actually the first one that we convinced Manuel to put on bioRxiv. So we're dragging him into the new universe, and this is the title, so you can go find it online. I think it's also linked on the event page. Cool. So, not working anymore. Is it gonna work though? Yeah, there we go. Okay, so let me give you a bit of an overview of what I'm gonna talk about, which will be in a couple different parts and pieces. So I'm gonna talk a little bit about the connection to AI. I mean, this is a neuroscience talk to some extent, but I think it has deep roots in AI and how we think about AI and how that those theories can then come in and explain some stuff in neuroscience. So this will be a high-level intro, hopefully bring everybody with me. All audiences, let's say. Then I'll talk about AI in practice. So even though I did my PhD here, I've been doing a lot of image processing in this postdoc. And so I'm going to talk about actually getting data, generating data. And that'll be a lot of AI as well. And then I'll talk about some causal and functional results in C. elegans. So this is now the more neuroscience side of things. I'm not going to do super fancy modeling. Okay, no neural networks here. This is mostly like the linear side of data science, but I think that's super important to do as a baseline, and then other people can follow up on that. So this is really like a baseline characterization there of this brand new data, that we'll get into and get excited about. All right, so let's talk for a second about history of AI. So AI started when they released ChatGPT a couple years ago. Okay, no. It started a long time ago, right? So you may have heard of this thing called the AI winter, where the AI kind of like lost a bunch of funding, and there's been a lot of oscillations in AI. This was back in a previous peak of AI. They published this book, 1981, called The Handbook of Artificial Intelligence, and it was all about these things called rules, and all about these decision trees where you explicitly enumerate, what you want your system to do. So this is tic-tac-toe. You can see these little stars, and there's an X, and there's an X, and there's an X. So depending on what you do, you make some decisions, and depending on what your opponent does, you make some decisions, and so on. So this is a rule-based AI. It's not just for games, it's also for birds, okay? And I think you can understand pretty immediately, what's going on. You have some kind of a type of bird. It's a subset of the category bird, right? This is a higher level hierarchical category. And then it has this relationship to this different concept called wings. And the type of relationship it has is described on this arrow, right? I think it's pretty easy to understand, and then it's pretty easy to generalize. You've got your favorite bird called Clyde, right? It is a robin, which is a bird, which has some wings. Okay, well, actually, Clyde has a nest, which is of type nest, and so on and so forth. So I think you can understand pretty immediately how this goes, how you scale this, how you generalize this, but also the problems you get into, right? I mean, like if you are manually setting up all these rule relationships, you're manually enumerating the categories in the world, it's a challenge. There are some successes. I really like this 1988 presentation in DuPont, which is a chemicals company. They said, what is an expert system? A program for capturing valuable knowledge and delivering it to the point of attack. Okay, I think maybe this feels like Nathan level language, actually. Nathan style language. But so people were using it in practice, right? In these companies, they had a lot of different sub companies, and they were trying to pool all their knowledge together, just like ChatGPT, right? You have a lot of, let's say, literature and science, and you're trying to pool all of that to get one system that you can ask questions. Right, so people were successful with this rules-based thing, but of course, as I mentioned, does it scale? Okay, so the analogy, and then to bring it into neuroscience, I would say is very strong. So there's been this classic research paradigm, which could be called reductionism, but basically that's just trying to go down to the basic parts, enumerate them logically at the level of cells and cell types and neurotransmitters and connections between those cells, and give those cells functions, right? And quantify these node-to-node connections, and then understand, what is the job of that neuron, right? Like in the same way, you're building a map, you're building a connectivity, and you're giving jobs to single neurons. And there are success stories here, right? So let me explain very quickly a little monkey experiment. First step, monkey looks at a center dot. Second step, it sees that these targets appear, but it doesn't do anything. Third step, it still doesn't do anything. It sees dots, which are moving either left or right, and then it has to make a decision. A lot of these experiments are focused on decisions, yeah. And it has to decide which is the direction of the dots. I think it's also, let's say, simulated in your mind. If these dots are very clearly moving, then it's easy. If it's noise, then it's hard, and people love this paradigm. Okay, so it's a decision-making paradigm. And you can model this using this beautiful little one-by-one circuit kind of philosophy and paradigm that I described. So you start out with random dot motion. You have some kind of sensitivity that individual neurons have to motion. And then you get some kind of noisy neural activity in basically single neurons or a small set of neurons. Then you can do simple mathematical operations that you can just write down. Basically, you just integrate up this motion, and then you get a slow increase in your confidence that the motion is left or the motion is right. Boom, you hit a threshold. Boom, you make a decision. Okay, so you can implement this, and people have experimental evidence that it can be implemented at the level of simple circuits. I can literally tell you this neuron does this job, this other neuron does this job, this other neuron does this job. I put them all in a network. They do this whole task. Okay, so it works. But yeah, right, does it scale? Can you actually build a whole brain? Can you build a whole organism out of this? You can do some simple decision-making tasks, but does it scale? So recently, there's been this big revolution in neuroscience, which I'll touch on several times, about collecting big data. There's a couple technologies that have made this possible, but on the theory side, people have been trying to understand this, and they've been developing basically completely orthogonal ways of analyzing data. So for this audience, I don't need to, dwell on dynamical systems too much, but fundamentally, there's this idea that you have a whole population, and that population is not doing everything. No, one neuron does not have a single job. They're all working together in concert to produce some kind of low-dimensional dynamical system, which has some interesting features like attractors. For example, in this flow field, you could think of these two attractors as decision 1 and decision 2 in this last little motion decision task that I mentioned. And so then you don't talk about single neurons anymore. You talk about spaces, subspaces, dynamics. Yeah. Okay. And of course, you can generalize this, right? It doesn't have to be single fixed points or 2 fixed points. You can get more complicated things. Again, for this audience, I think it's not that complicated. But I will then just mention that there is evidence of like ring attractors that are being implemented in Drosophila, for example, which are fruit flies, that help in navigation. Like this exists in the brain, right? And then like limit cycles, I'll show you something that looks like a limit cycle, and I'll try to talk about that, but don't have like, I'm not modeling it as a limit cycle, but it looks like one, certainly. And taken together, I would call this, and I'll call this several times during this talk, this manifold hypothesis, which is 2 pieces. That there's low dimensional subspace within your space of all of your neurons that is doing the thing that matters, that's doing the computation, as well as some kind of dynamical system understanding that gives you understanding of what's happening in this low dimensional space, right? It's not just randomly doing stuff. It's doing something with a clean flow. It's doing something with fixed points. Okay. And there's some really cool success with this manifold hypothesis. So this is now a different monkey task. People love these monkey tasks. And you have it starting out here or starting out here, doesn't really matter. And then by some cue, doesn't matter, it's reaching to some angle. It's reaching, in this case, down a little bit, or it could be reaching up, or it could be reaching to the right. It's reaching at some angles, and you just have it do a bunch of different reaches, and then you image some kind of motor cortex or something to try to understand how are these reaches, how are these motor programs encoded. And you can do things that are at the single neuron level, and it's like a little hard to understand exactly what's going on. Maybe you could decode something from this one neuron. It starts the movement here, and then it goes up. Okay, that's kind of interesting. But the story that people tell, and one of the seminal papers in this field in 2012, they did their own special algorithm for decomposing this into a low-dimensional subspace, and they found this really nice rotational dynamics as the monkey is kind of doing this reaching, and this red and green encodes like angle, and you can kind of see if I pop this up again, you can kind of see that they're coherently following some kind of interesting dynamical bundle or dynamical flow. Okay, so I'm not going to get into the details of this paper too much, but just to say everybody got really excited, and then it kind of like set up this explosion of this style of analysis in neuroscience. Okay, but there are some significant gaps in the whole research program of the manifold hypothesis. One thing is that maybe you get these beautiful low-dimensional picture because your behavior is so simple, right? In this case, the monkey is literally doing this, these reaches. It's not doing a behavior that's high-dimensional. It's doing a 2-dimensional behavior. So maybe it's not actually that surprising that you get a 2-dimensional pretty picture, right? Also, too few neurons were imaged, perhaps. In this case, especially in 2012, you have 100 neurons out of like 1 million in one brain region, which is, like 10 billion then if you expand it to the rest of the monkey. So like maybe you are missing a ton because you're so dramatically subsampling. And this has been really hard, although people are working hard to get more neurons, on monkey level, it's very hard to, like, you can't image the whole brain. You just can't do that, right? So dramatic subsampling in these big animals is the state of the art. Okay, but another question, if I really believe the strong version of this statement, what is the rest of the brain even doing? Why do I need a million neurons if the whole brain is just two-dimensional? Why do I need that? Why would evolution do that? So we'll try to answer some of that today, or at least give a proposal. And another thing, which I think is really important for this audience, neuroscience is funny because there's been this big influx of computational people, especially from physics. And they do one of two things. They either take really cool data and they just do PCA, which is kind of the thing that I'll show you today. Or they make up a completely custom method for each paper. And then it's very difficult to know, okay, well, what are the problems or pitfalls with this method? Because you literally just developed it for this paper. Like there's no math, there's no proofs, there's no theory behind this. You know, the simulation people have not tested this. You didn't do this on the Lorenz system. You literally went straight from, you know, I show you some pretty picture that I believe is happening, and then I use my novel method on my real noisy data. And so it's quite difficult to understand what are the pitfalls of these methods. And that's like super important. Yeah. Another thing is like intrinsic versus extrinsic factors. So, you know, to some extent in this exact example, it's known that there's like intentionality behind it, but it's in general very difficult to disentangle. You know, if my muscle is doing this little reach, there's all sorts of sensory information which is going to be correlated with this. And it's very difficult if you just have neuronal data to disentangle, is it actually coming from the brain, this two-dimensional structure? Or is it just sensory measurement, which happens to be two-dimensional, because the sensory measurement and the intention are going to be correlated. I'll get into this a little bit more. Then the last thing, and I leave it for last because I'm not going to talk about this too much, but I'd love to talk with people more offline about this. You know, is this really a state space? And what I mean is this two-dimensional space actually describing or well described by a dynamical system? Is that actually what's going on? Because that's two different statements. The brain is projecting down to a low-dimensional space, and the brain's evolution in time is actually simple. These are two different things that's, yeah, won't have too much time to talk about, but I've been thinking a lot about this, so I'm excited for brainstorm. Okay, and I showed you a bit of monkeys, but it's not just monkeys, right? So manifolds or something like this have been observed across species. So this, for example, is a mouse running on a little trackball in virtual reality, as is the fashion. And you can image, you know, 10,000 neurons, still dramatic subsample of the brain, but you can image 10,000 neurons and you can see that there's this kind of like coherent structure that's happening alongside this green annotated behavior, which is like the running behavior. And the first PC component, you know, always do PCA. You have to have PCA in your neuroscience papers, otherwise you get chased out. So, you know, this is like a science paper where they did PCA and other things. And you get the dominant mode is really correlated to this behavior. And you can describe this, although this paper doesn't do it in this way, you can describe it using this low dimensional perspective. And in flies, you know, more than 1,000 neurons, again, dramatic subsampling, you get this coherent structure. I'm not going to show you any quantifications. I think it's really obvious that you get this coherent structure that's loosely related to this running behavior, again, on a ball. And in C. elegans, now this is back to the lab that in the world that I'm familiar with, there's this classic work on immobilized animals where you can see this really nice, beautiful, coherent structure in the brain that corresponds to fictional behaviors because it's actually immobilized. And you can see, again, the first PC component shows this really nice up and down, up and down, up and down structure. Okay. Okay. But, you know, as I just mentioned, there are some gaps in the overall manifold hypothesis, but there's even some gaps here. You know, why are all these guys correlated to ongoing behavior? Why is that the thing that's like encoded at PC1 level? And what are the actual origins and functions? Like, let's dive down. Let's really understand, what is this actually doing? Is this just epiphenomenal? Is this really real? Or is it just, you know, suffering from some of these problems that I mentioned before, right? If we go to more complex behaviors, do we still see this? If we go to more neurons, do we still see this? And so on. All right. So, worms. They're beautiful. Okay, they're absolutely, like, look at this. Look at this. Look at this little rainbow. Okay. But it's not just beautiful. It's really beauty that gives you scientific power. Okay. So they have 302 neurons, as Nathan mentioned, and you can image more or less all of them simultaneously at single cell resolution. But you also know the connectivity between all these neurons, and they each have a genetic destiny. Okay, so each one is genetically like known, everything that it expresses at the single cell level. And so that means that you can build a very powerful tool recently. You can build a colorful map that uniquely identifies each neuron based on a color, a combinatorial color code of expressing. I think in this case it's 5 different colors, like a green, a blue, a red, and like another red. You get these pretty colors, and then that allows you to identify every neuron, and then you can compare the spaces. You can compare data across animals. That's very, very important.
Wait, you actually genetically program those cells to fluoresce?
Genetically do so much. So you genetically just absolutely mutate the heck out of these animals. And these guys are like super sick, honestly. They're not happy. But they're super pretty, yeah. And then you also mutate them so that they will fluoresce. Okay, so this is now, you can see it's sped up, but you can see that these coherent patterns are now like lighting up across the brain. Some neurons going up, some neurons going down. These coherent patterns exist, and they're just super great. They're super great. And really, this is the first system for whole brain imaging. There's some stuff that you can do now with larval zebrafish, and another couple fish are coming online where you can really image the whole brain. And to some extent, you can do this with fruit fly, at least the whole like central brain. But C. elegans was really the first to do it, and there's a huge wealth of data, and it just is begging to be analyzed, really, for sure. Okay, so let me tell you now about where my project started. So when I came here 5 years ago, people had already done this. People had done immobilized animals. People have all these beautiful little manifolds, all these beautiful, pretty pictures, but it weren't actually doing any behavior. So what they wanted to do is get real behavior and real data simultaneously, and then all their dreams would come true. That's what the experimentalists would say, right? This is the holy grail, yeah? Real behavior and so on. So you don't have to know too much about C. elegans, but just so you know, here's what they are. Here's what they look like. And they've got this sequence of behaviors.
They're like 20 feet long, right?
They're 20 feet long, right? They don't even fit in this room, actually. But you still need microscopes. No, they're about 1 millimeter long. And so you need a lot of microscopy to get all of this to work. And they go through this motor pattern. Okay, they do this forward crawling, then they transition to a reversal, and then out of that reversal state, they go into one of 2 turns, and then they return to forward crawling, and then they just go on their happy way, and they do this. So already I can kind of like maybe convince you that this would be perhaps well modeled by a limit cycle, because there's literally just this cycle of behaviors. No neurons yet, yeah? But this is now what I was confronted with, 5 years ago. This is the data that they had, and it's beautiful, right? You get the single cell resolution. Yeah, if we could turn that. Yeah, look at that. Look at that. So you get this two different reference channels, and we have this in 3 dimensions across time, so 4 dimensions at 3.5 hertz, and you get this really beautiful. Just let me start you over again. You get this really beautiful set of videos, but of course, videos are not any of the manifold stuff that I saw before. And the lab said, maybe it'll be 6 months, maybe it'll be 8 months to like get time series out of this. So 4 years later, I did a ton of image processing, which I'll talk about a little bit next. But first, I've got to thank the actual experimentalists who did all this, like a full-time microscopy guy to build microscopes and debug everything, and 2 wonderful experimentalists that it's been absolutely amazing to collaborate with. Okay, so on my side, right, I had to build a pipeline, not just a pipeline, but a pipeline that people who don't know computers can use and run and be happy about. But fundamentally, this is 2 problems, okay? This is segmenting and detecting all of these neurons, and then it is tracking. So it is associating them across time. And so, long story short, as you do in science presentations, you compress years down into a slide or 2. We use neural networks to solve it, okay? So I'm not going to talk too much about segmentation because that's relatively easy to get, you know, an 80-90% accuracy. So I'm not going to talk too much about that. We annotated a bunch of data, we trained our network, boom. Here's the citation. But the tracking, that's more interesting, and let me talk about that for a little bit. So we start with detected objects. That's segmentation. I'm not going to talk too much about that. And just imagine we have 2D instead of 3D. So time is here, these little slices, and we have all these little objects. We don't know who is who. We don't know which objects are the same across time. We just have objects. Excuse me. Okay, so what I did is I matched sets of points. So I took some really cool work that has a cool name. It's called superglue from the computer vision worlds. They like their super names, right? Just like Cindy and Shred and all these cool names. So there's a superglue network, which is a graph network with attention. Basically, it was like just before transformers, but it's effectively a transformer on a graph neural network. And it tries to match sets of points between different views of an image. So I think you can imagine that this chair, you know, is the same chair over here, but it's like rotated. Yeah, this is like a common problem in computer vision. And this is very similar to what we want to do, right? We have sets of points, which are actually neurons, and we want to match them across different views. Now we have non-rigid motion. We have the body that's squishing, and it's really non-trivial. So we're not just doing a simple rigid alignment, which there's tons of work on rigid alignment. We really have a more complex problem. And unfortunately, all that beautiful rigid alignment work just didn't work at all. It would have been great if it did, but it didn't. So what you do is you build up this two by two matches into short tracks, which are called tracklets. And you can train a neural network using annotated ground truth to do this pretty well. And I'll just say that I actually had to build a graphical user interface, and we hired a bunch of undergrads. We had a little army to do ground truth. It took like 3 months to I don't know, 1,000, 2,000 like human hours to really get this ground truth. And so that was a really fun experience of hiring those people, making sure it worked, making sure the back end and the front end worked, and making sure it worked on Linux and Windows. And so it's a fun time. But then you put all this together, you stitch together these short time guys into full time tracks. Okay, so that's the data that I'll show you comes from this pipeline. And one of the main benefits is that this is actually pretty easy to correct for the humans. In our tests, correcting these tracklets versus correcting single points was about 100x speed up, because you have some help from the computer to do this. Okay, so it works well, but you know, this is supervised learning. As I said, we took that like thousands of hours to train it on our microscope, on our data, on our settings. And you know, if somebody wants to download this from another lab, which of course you want to do, because it's science and you want to be open, it's not going to work. Like, it might work a little bit, but it's not going to work. And this is kind of a dirty secret in the field. We have a couple collaborator competitors like at MIT and so on, and they have their method for doing it, works on their data. It's not going to generalize anywhere else. And so what we would like to do is have some method that works, but also somehow absorbs information about your raw data directly without a ton of manual annotation, in order to match to that data, match to that laser, match to that, you know, resolution, and so on and so forth, or future microscopes, right? So of course, this is a huge field of study, and basically the answer in the computer science world is self-supervised learning. And so I'm gonna give you like the shortest introduction ever into the next project that I'm working on that I'm really excited about, but there's no preprint yet for this. And I'm calling it Barlow track, so I have a fancy name for this. Okay. So what I'll show you is from a paper called Barlow Twins, and it's one architecture for self-supervised learning. Okay, so this is not the only way to do self-supervised learning, but all of these different architectures have a lot in common, which I think I'll get with this. And most of these architectures basically start with some sort of image. I'm not going to talk about words or natural language or anything. Well, you start with some sort of image and you distort them in some way from some, you know, transformation space or some augmentation space that I'm calling little t here. And then you have two copies of the same network. They're twinned networks. And you build basically an encoder structure. You can have lots of different things. Here they have a bottleneck that's not really necessary. And you get this latent space Z, okay? Again, going, so two different distortions, let's say rotations, two different rotations, two different croppings, something like that. You build a latent space, and then what do you want to do? You want that latent space to have some special properties. And in this case, what Barlow Twins does is it calculates the cross correlation across feature dimension of your whole batch. And it wants that cross correlation to be diagonal. So what that means is that different augmentations of the same image. Yeah, let me give you just to be a little concrete, we're going to have neurons or a little crop around a neuron. So neuron and its neighborhood. We're going to rotate it or otherwise distort it in two different ways. And we want the representations to be invariant to those transformations. So that's the diagonal term being close to 1. And we want our feature space to be expressive. We want our feature space to be high dimensional. And really specifically, feature I and feature J should not be the same across your input set. Yeah.
This autoencoder that you're showing twice is the same weights, right?
It's literally a twin. It's the exact same network. Yeah. So you want the same network to be able to digest two different views of the same image and give you approximately the same point in latent space back out. Okay. So there's a lot of tricks to get this to work. There's like one key trick that I don't quite have time to go into, but then what you can do in our case is just directly cluster that latent space. So in this case, I'm showing you a t-SNE embedding of an HDB scan clustering, but fundamentally, you can use a lot of different clustering algorithms. You can, and I'm not actually using this state space or this t-SNE space for clustering. I'm clustering in a higher dimensional space, but I give it all of the neurons across time. And if I do it well, then it will cluster all those neurons across time into the same cluster. And that's tracking. That's unsupervised or self-supervised tracking. No ground truth needed. This actually works in preliminary data across species. So not just C. elegans, but people are really trying on the biology side to get new model organisms to come online. And we have this again, preliminary, but it's working well. And yeah, I'm just really excited about this because this is now finally, you could actually, as a new experimental lab, you could take this, you could download this, you could actually get data on your system without, hiring a computational postdoc for 4 years to make it work. Yeah. Which is the state of the art, right? Yeah, and I really want to emphasize this point. Like, I think you guys are working on some beautiful, clean data, but like in the experimental world, it's years to clean anything up. Yeah.
So
Yeah,
you're calling what your algorithm? Barlow track?
Barlow Twins goes to Barlow track.
And it's gonna be pi Barlow track?
It's going to be pi Barlow track probably.
Okay.
Yeah.
Pip install that.
Pip install that. It's going to be great. Yeah. So that's my summer. Yeah.
Okay.
That's what I'll be getting to work and writing up and send it to you.
Awesome.
Yeah, for sure. Okay. So an intermediate summary, and then I'll stop talking about, I see a quick question from the chat. Are the rotated images only rotated or also skewed? In principle, you have a space of augmentations, and that's going to be basically relevant to what you think the augmentations are that are actually in your data across time. So you don't need the distortions, like you don't need to build a model of what's happening to your data across time. You don't have a generative model because it's non-rigid and it's crazy, but you want it to be similar to the distortions that happen across time. So if your animal rotates in Z, because these are actually 3D, then and you want to skew across that dimension, then yeah, you should put that augmentation in for sure. Okay, so intermediate summary, we've got whole brain, freely moving, single cell tracking and traces, and self-supervised learning takes this to the next level where you can really do this across labs, really actually generalize it. Okay, but what do we do? And I will show you some baseline quantifications, and then I hope you guys download the data and do your own stuff. So back to the analysis, back to manifolds. So let me just remind you a little bit. One of the problems in this manifold space is that perhaps the behavior itself is two-dimensional, and therefore all your results are kind of artifacts. Boom, we have freely moving animals, truly doing a complex repertoire of behaviors. Too few neurons imaged? Well, I've got a whole brain for you. What about higher dimensional activity? We're really specific. I'm going to specifically dive into some analysis there. And I think that there's a lot to be learned from diving into this, even if you also do a manifold-based analysis. Artifacts of complex analysis methods. So for this, I'm really going to stick with linear stuff, right? I'm really going to stick with the simple stuff because I think that's a super important baseline. Doing more later is absolutely amazing. Yeah. Intrinsic versus extrinsic factors. I can actually, in C. elegans, do some really cool causal work, really understand the causality of certain neural activity patterns and disentangle this to some extent, okay? Is it really a state space? Again, let's talk later. Okay, so boom, here's a data set. And this is like one of our first data sets from freely moving animals. And you see this coherent activity pattern. You see this beautiful begging for PCA little data set, okay? And now we have real behaviors to go along with that, okay? Forward moving, reversal, ventral turns. We have real behavior. It's really correlated with real behavior. Amazing, okay? Right? Really, truly holy grail of neuroscience. And then PC1, also PCA is also the holy grail of neuroscience. I can't. It's the gift that keeps on giving. Okay, so, you know, but we can see that qualitatively, it looks like PCA is following this behavior. PC1 is following it, PC2 is following it, PC3 is more complex, and it's following the speed, yeah? And then we can put this phase space together and really get this forward kind of fixed point that then zooms over here to a reversal, possibly fixed point, possibly slow point, and then just kind of slowly collapses back here into the forward fixed point and so on. It does this really, really nice distinguishing of the behaviors in this absolutely the simplest state space ever. Okay, and what does this look like live, right? So you see that the worm is moving forward and the brain is sitting here in the forward fixed point, and then it zooms over here to the reversal slow point as the worm does the reversal, and then it chills out here for a little while, and then it terminates the reversal and it's now turning, and then the brain also just slowly, noisily, but slowly decays back to this forward fixed point, and then it chills here for a little while and it continues on. Okay, right. So this is like, we were super excited to see this, and there's only a couple labs in the world that can get this kind of data. And so we always like to say that, yeah, we're neck and neck with Harvard and MIT. At the University of Vienna, that's very important. In Europe in general, it's very important to say, like, you're competing with the Ivy Leagues, right? You got to say that. Okay. But so I showed you a pretty example. I showed you a pretty video, but you know, let's talk a little bit more quantitative. It's still a linear method, but let's talk a little bit more about what is the relationship between behavior more broadly, not just colors, but behavior as time series and neuronal time series. So on the left, we have neurons. I've compressed it a little bit. And on the right, we have some kind of behavioral metrics. So we have what's the head doing? It's kind of bending around in various angles. What's the body doing? It's also got kind of like increasing curvature transiently. What's the speed of the animal? It's got this interesting dynamics. And the question that our lab really wanted to ask is, again, going in this low dimensional direction, you know, is there a latent space that's shared between these two things, not just some kind of PCA unsupervised latent space? And for this, we used like off-the-shelf, simplest starting point, canonical correlation analysis, which tries to find a latent space where you have a projection from this side, a projection from this side, and you try to find the dimensions that are the most correlated. And very interestingly, and we can quantify this across data sets, we actually get nearly identical state spaces using CCA, so using a method that's aware of behavior, and PCA using a method that has no clue about behavior. So you really get that the brain really, in its most highest variance modes, is caring about its instantaneous behavior, just like the mouse and Drosophila that I showed you earlier. But we had this key question, which I think is not asked enough in the worm field, at least, which is, let's compare this to a bit of a null model or a bit of a simpler model to understand this question. Are these continuous behavioral metrics really required? And so we replaced these continuous metrics that I showed you with just discrete metrics, you know, ups and downs, one hot encoding of the behavior state, and we get the same effectively state space. So this mode 1, if you do the dot product between these different ways of calculating it with CCA and PCA, almost identical. Absolutely, the first mode captures this state transition. The second mode is a tiny bit different between the methods, but is across data sets very, very, very similar. So low-dimensional population activity is consistent with this discrete state representation. So we're not going to say that like that's what the manifold is doing. We have no causality at this point, but the experimentalists really want to say that the manifold is encoding the state, the high-level state. And I'm going to be a little bit more cautious in my statement and say it's consistent with an encoding of the high-level state. Okay, so now that we have this high-level state is consistent with encoding a high-level state, we want to understand those other gaps that I said in the manifold hypothesis. You know, like what are the origins and functions of this manifold? What is it actually doing? Where does it actually come from? Like, let's step away from correlation methods. Let's go deeper. Yeah. Okay, so one hypothesis that's really out there in the field is that this could correlate, or this could be coming from intrinsic motor commands. The intention of the animal, yeah, could be encoded here. And then, okay, I need to hurry up. Okay, the intention of the animal, or it could be coming from sensation, because as I do behaviors, I create sensations that are correlated with those behaviors. Right, so I need to be able to disentangle those, and you can't really do that with just one data set. And another hypothesis is that it's actually doing some interaction with higher dimensional activity. Low dimensional activity is somehow organizing high dimensional activity. Okay, so let me compare on the left a freely moving data set and an immobilized data set. So qualitatively, there's a lot of similarities here. You know, there's longer states here on the right than on the left, but qualitatively they're very similar. But we can't just stop at this qualitative level. We want to understand, are these actually the same neurons? Is this actually the same neurons doing the same job and having the same intentions, independent of motor execution, or is it just a qualitative thing? And the way to answer that is, are these the same neurons? Which you can mention, or you can go back to the idea that we can identify neurons across animals. They're really the same neurons. And if we see that the same neurons are participating in the same way, then we can understand that that neuron's activity is independent of motor execution. Okay, so here's a long list of names. Please don't be afraid. Okay. You don't have to understand too much, except to say that there are many neurons. There's a whole community that are active during reversal. And we can show this simply by the PC1 weight. They have this positive weight of PC1, and they're doing effectively the same thing in both conditions. They're doing the same activity pattern, regardless of whether it actually occurs or not. Okay, really, it's an intrinsic pattern in the brain. It's intrinsic dynamics. Maybe it's a dynamical system. But there are these really interesting exceptions where they're not doing anything in the immobilized case, but they suddenly jump up and they have this really strong correlated activity when the animal is actually moving. There's a whole list here, but just to say there's also a forward community that is doing something similar on the opposite side of the behavioral spectrum. And putting all of this together, we can quantify that about 63% are like intrinsically motivated. They're doing the same thing in the immobilized and the freely moving case.
These neurons that are showing a difference between immobilized or freely, how close are they to sensory neurons?
Yeah, so let me zoom in on these two. So what do these differences mean? So one of these is actually a really nice, well-characterized and known sensory neuron. So this is a time series trace of the activity. As it moves backwards, we get up to a peak here, and then it goes boom, the nose. It's actually when the nose is colliding with the body. It actually has an additional peak in addition to being reversal correlated. And we can see the second peak. I think you can go 3, 2, 1, boom, nose collision again. Okay, so we were super excited when we saw this. And it turns out that we've actually published a whole paper on the lab, just diving in to this single neuron and the pathways there. And it turns out that the CO2, the breathing out, the gases of the worm are smelled by the worm as it reverses into its own gases. And so you can do a simulation of the CO2. I could do a simulation of the CO2 at the nose of the animal, and it looks just like the reversal. And if we had a nose collision in this little video, you would see another spike there. Yeah, so it's actually just, you can never tell this from like pure modeling methods. It's correlated to the behavior. Causality is completely different, right? It's extrinsically generated. And it's not just one neuron. There are several others. This one goes up during reversal. This one goes up during forward. And yeah.
Yeah, I guess like following the question, you have this clear sensory neurons that are showing this
Yes.
difference between
Yes.
localized. One synapse apart from that, like you see the same effects or like it just disappears.
It is largely localized to the first level. In our data, yeah, it's largely localized to the first level sensory neurons, these dramatic differences. But there are some interesting, noisier examples of differences that people are diving into at the second level there. And another cool power of worms is that you can actually remove this ability to smell this entirely. You can remove the oxygen and CO2 sensing pathways, and you can see this effect go away. So you really have a lot of causal control over this animal. So putting all this together in a pie chart, we have a ton of neurons, really a lot of neurons that are shared between freely moving and immobilized. This is like an intrinsic motor copy or efference copy. And we see another further like 9% or so that are sensing just this one pathway. There could be other sensory stuff that we didn't fully characterize, we don't fully understand, but just this one type of sensory pathway gives you a ton of behavior correlated activity as well. Yeah, cool. So next hypothesis is that there's something going on besides this low dimensional manifold, because there's more complex behavior going on than just like this two-dimensional behavior. Let's try to understand that and its relationship to the manifold, if any. All right, so here is one neuron. Here's the activity of that one. I want you to understand two pieces of qualitative information about this neuron. It's oscillating, and it's kind of dying off during the reversal. Then it comes back up, oscillates, dies off. And we can mathematically decompose this really simply using PCA into this shared component, shared with the rest of the brain, manifold component, if you will, as well as the subtraction of that, the residual component. So we just have this oscillation, and then we got rid of this modulation. And this is super interesting, especially to the experimentalists, because this is actually correlated to a behavior. This is correlated to the actual undulation of the animal. This is correlated to the muscle activity at this close by this neuron. But it's not just doing its own thing, it's also talking to the rest of the brain. Okay, but what we want to understand is there a general relationship, mathematical relationship, not just a pretty picture that I show you, coordinate between the red and purple guys. So one way to do this is to do a triggered average. And what I mean by that is you take the same posture across your video, and you just say, okay, what is the activity of that neuron whenever I hit this posture? And during the forward state, it has this really nice correlation. And during reversal, that encoding goes away. This is something that biologists love to do. They think this is interpretable, so you always have to do this first, just like you always have to do PCA first. But we want to go beyond that and build a model. So I've tried a couple different ways of explaining this model, and haven't quite set on like a really good way to explain it. But fundamentally, it's a linear model where you just have neuronal activity on the left side and a series of behaviors on the right side. Okay, that's nice and simple. You just get some sort of weight matrix that combines these behaviors in order to attempt to explain this. This is your behavior only model. And if a neuron is encoding behavior, that's kind of exciting. I don't need a manifold for this stuff. I just have behaviors, and then I have neural activity. No manifold involved. What we really are interested in is neurons that display the following property. And we like to call this, the experimentalists like to call this a hierarchical model, but I think it's easier to understand it's a state-dependent encoding, or state-dependent activity. So we put a sigmoid onto the manifold to modulate the behavior, and then create a nonlinear model that explains your time series. And if this one is better, then this neuron is displaying this modulation feature. So you really have this interaction between this low-dimensional global thing and the behaviors. Right. So this hits this gap in the manifold thing where it's a manifold hypothesis where it's not just this, it's this interacting with behaviors in order to explain the neuron. And long story short, we can apply this across neurons. And I actually did a Bayesian multi-level model to quantify the differences between, inter-individual and so on. But suffice it to say that you get a bunch of neurons that display this property. And if they are above this x equals y line, you get an improvement by adding this hierarchy term into your model. And you get some that are dramatically improved and some that are, improved only a little bit. And I'll just say really quickly, we have motion artifacts, so you have to carefully control for that. You have to always control for motion artifacts. But it's not just a bunch of neurons doing their own thing. You actually have this really interesting phase relationship between the neurons because you have this undulation. These are sine waves. So I can plot this in polar coordinates. And we see this really clean phase relationship between a bunch of different neurons that is very consistent with the literature and some of which are novel. Okay, this is the example that I showed you. It's like basically the strongest one. Obviously, that's what I showed you as the example, like the best one. Okay, and just to show you all of this put together into a pretty picture, this is our little favorite neuron. It's red in this little banana posture. It's blue in this little banana posture. And the other neurons, this one is blue, this one is red. So red means active, blue means inactive. And as the animal changes, you can see this red now goes to blue. And so we can see in real time across the brain, this instantaneous posture, this high dimension, relatively higher dimensional posture being encoded in addition to the highest level state. Okay, boom, conclusion time. So a lot of prior work went into manifolds and reaching monkeys and doing simple tasks like this, and they measure one specific brain region and whatnot. But we've got the whole brain. So we see that this concept, this manifold concept really generalizes well in this system to organize behaviors across timescales and across the entire brain. So not just preparation and execution of like one thing, it's actually organizing the entire repertoire across, like hours of the animal. Okay, but it's not just organizing that high level thing, it's also talking to the multi-scale behavior that we observe in this animal. So you've got these states, you've got this these more specific, more detailed motions at a faster time scale, and this guy is organizing this guy. And we think that we can find signatures of this in other animals as well. We think that this can generalize outside of worms, and I don't know, maybe even build some cool AI systems out of it. I don't know. And then causally, we can also do some really cool work in worms to be careful not to overinterpret our data. We've got a lot of beautiful correlations, but you want to understand that at the causal level, not just the correlation level. Got to thank the lab and a really, really excellent undergrad who did a lot of the oxygen work. And well, that's me.
All right. Floor is open for questions.
I've got one question here from the chat, just so I do well by them. Is it true that you think of your neuron data as an autonomous dynamical system? I absolutely do not think of it as an autonomous. My PhD work analyzing some similar but immobilized C. elegans data was really all about discovering control signals or discovering unknown pieces, non-autonomous parts in the data. And I think that's really the way to go, because I think that there's sensory information coming in. There's even though you have the whole brain, there's faster timescale stuff that's unobserved, and in practice, these transitions are just not actually predictable so far. Like I showed you this video. Won't go back to it now. But these transitions are not actually predictable. It really does not seem like it's an autonomous system, except for certain points in time. Sometimes it seems autonomous, but other times it seems really obviously like kicked or actuated in some way. Yeah.
So is it possible that there's these switching neurons, I can call them, like the ones that are sort of responsive to the behavior first, that it's like at a neuron level that the switch happens between the forward and backward behavior?
So let me answer
Or
that.
is it more like a global switch in that all of these residuals like determine the switch, or maybe the switch is more determined at a neuron level, it just kind of goes on and off for different behaviors.
Yeah, so I mean, the neurons that determine this forward-backward switch, there's like a whole community of them, and they really all come on simultaneously, and they all come on. Some of the fastest timescale things that happen is the initiation of reversal, and that's not predictable. So there's not like one friend that comes up first, and then it like brings everybody else with it. There's not like one, oh, I'm gonna prepare, and now I start reversing.
Maybe there's
That's,
different modulation between them.
that's absolutely possible, right? There's quite a bit of invisible communication that's happening. There are these latent variables. It's not just direct neuron to neuron communication. There's also like spreading, diffusive communication that happens, and that's another latent variable that we're not measuring for sure. It's very plausible. Like there are lots of explanations why we couldn't predict it, but
And the like like a year or so,
fine.
and like he was just nailing the peptide stuff, like if he had peptides.
Yeah,
communicating in that.
so,
I don't know what you're talking
look,
about.
I've seen his talk on this, and it's fundamentally impressive work, but the devil's in the details. The data that they have are very good, but they're also very noisy. Like they do this actuation, and then they measure, the responses, but the responses are happening 20 seconds later, 15 seconds later. The way they do the actuation is this really interesting, super interesting, but very potentially unnatural type of like actuation profile. So it's not clear that you're really actuating things consistently. So yes, the neuromodulation thing is one explanation for some of the things they're seeing, and it certainly makes sense in certain cases, but it's not, I wouldn't generalize as strongly as they're generalized. Let's say it like that. Yeah. But I could talk all day about that. Yeah.
I have a question about the like self-supervised learning.
Yeah.
method that you
Yeah.
have. What kind of loss were you using for your Barlow twins model?
So yeah.
I think it's like in the hot field right now. It's curious.
Yeah. So fundamentally, the Barlow twins paper itself, like the original paper, has this diagonal loss function, where you have the on diagonal term you want to be ones, and you have this off diagonal term that you want to be zeros. And so that's the original loss function that, as presented in this paper, is this, and that encourages these two things, right? Each so you have like a lambda to balance, which then you want more, right? The high dimensionality more, or the invariance more. So you have one lambda term. In practice, for the video tracking task, it's also very important to do the transpose of this, so that you get two additional loss terms, not across the feature dimension, but across the object dimension. And then you have 4 terms in your loss, where you have but it's just a doubling of this. So it's conceptually the same thing, but now you have 4. I could, yes. Yeah. Does that help?
Yeah, that helps.
Okay. Yeah.
Hey, I'm not familiar with the actuation work that you mentioned earlier, but in terms of like just doing, let's say a simple system ID, on like you now discovered that there's like a mid-cycle, there can be a low dimensional systems now. Like, what is the step to like basically push the system to identify like an input-output relationship, or a very simple, like a linear dynamical system? Yeah. Like, is there a next procedure, like we'll just push this system, let's see the output, let's figure out A and B matrices?
So when you say, I guess there's 2 parts of the question there. When you say let's push the system, you might be referring to actual experimental interventions. Yeah. So this is the direction that a lot of people are going in, like the lifer work mentioned earlier, where you're pushing one neuron at a time, and you get this kind of causal understanding of the system in that way. We are attempting, and it's the project of a current PhD student, to do live, like closed loop control of arbitrary patterns of neurons. But that's absolutely, it's not really a capability that people have to do simultaneous imaging and actuation. It's very difficult, except in this really interesting pathway that the lifers have developed. So that's something people are trying to do, right? They're trying to understand, is that, if I push it this way, does it like decay back? Is this really a fixed point? This is something that people are absolutely actively really
Yeah, the
excited about.
latter thing you mentioned, basically like you have a good enough feedback or closed loop simulation, then you have like an internal model essentially. like a
With
good-
simulation.
Yeah,
Yeah, so people are also building lots of different simulations. I'm going to hear about how Shred solves all the simulation problems
In the world.
in the world. In the
Oh.
world. But people have, the mobilized version of this. There's been, I don't know, a dozen modeling papers doing different things. I did a DMD based thing where it's a single linear system.
Yeah, I was gonna wait till you mentioned your DMDC.
Yeah,
work on
I
this,
know,
like,
yeah.
Cause Nisha's
Right.
working on this, so you should.
Okay, we should talk. Yeah.
Yeah.
So I did a DMD-C model where I discovered these control signals, and then fundamentally the dynamics were linear. You've got a paper, right, on discovering an ODE that has actually multiple fixed points as well as some controls. There's another former PhD student in the lab who did like a sort of handmade ODE system with control. There's a ton. There's this hidden Markov model, switching linear dynamical systems. Like there's a ton of things, but there's a lot of different ways to generate this data, but these are correlational methods. Like you're not gonna really, although maybe Shred will solve all the problems, you're not gonna generalize out of your distribution, right? So you're not gonna be able to cleanly predict what will happen if I do a novel perturbation off this manifold, right? Like you're not going to, in general, discover the true causality of out of distribution perturbations, except with Shred.
No, oh, it's an example, maybe.
Yeah. But this is I mean, that's a long answer. And just to say, like, people are super excited about this and people are working really hard experimentally on this. And it's going to happen. Like in 5 years, that data is going to be there. And it's going to be downloadable and it's going to be really cool.